Big round of applause, please, for them. Right. Next. Now we're gonna blow your minds with some real science. So I am absolutely delighted and want to thank the Royal Society of Biochemistry for providing us with tonight's expert speaker. Charlotte, please come take the stage.
And one clip over it. Oh, one of these as well. Fantastic. Thank you very much for having me. Hello, everyone. Hello. How are you all feeling? You feeling good? Yes, I'm feeling good, and I'm so delighted to be here at the 18th Sci-Fi Festival. It's just an absolute pleasure, really. And I have been invited by, oh, this is a lethargic clicker. I've been invited by, yes, the Royal Society, the Royal Biochemical Society, to be here today. So thank you very much for having me. And I'm really excited by Chimera. I really am. It's a fantastic film, and I'm really excited for you guys to watch it. Can I have some hands up? Who is a scientist in the room here? Ooh, yeah. Okay. Hands up. Who's a microbiologist? I've got a microbiologist. No. Geneticist? Oh yeah, me. Geneticists? No. Epigeneticists?
More granular.
Biochemists? Physicists. I forgot about physics. Oh, that's a nice one, isn't it? Oh yeah, and chemistry. I forgot about that one as well. Chemists? Of course there isn't. Yeah, they're in the lab. My brother's a chemist. He is currently in the lab. It's not a joke. That's where he is. Yeah, so this is actually a picture of my lab. Like I said, oh, there we go. Should we come back? There we go. I am a geneticist and an epigeneticist. So when everyone laughed at epigeneticist, I was like, oh, okay. And I'm a microbiologist. So genetics, genes, epigenetics, I'll come on to that. Microbiologist, I work with the small things. This is my lab behind me where I've been working for about 5 years now. And lots of the things that actually occur in the film Chimera we do in this lab. Some of them we also do not do, but quite a lot of them we do actually do. So, yeah, I mean, for me, mutating things is a daily occurrence. Sometimes on purpose, sometimes by accident. I did occasionally make something that I did not know what it was. And then, you just have to kill those things. It's literally not a joke. You just kill them. You look at them for a while and then you kill them. So yeah, that's what I've been doing for a while, 5 years in the lab, working on genetics and epigenetics, but I am a massive sci-fi fan. Been mutating things, I've been extracting and purifying protein, which is fairly boring, and I feel sorry for anyone that does that if you are a biochemist. Chimeras, we don't make them here in this lab, because you do that in the animal house. That's somewhere I've never seen. You do smell it, but you don't see it. That's where all the mice and all the animals live, and we do have to make chimeras for a lot of our experiments. So first of all, I'm going to talk to you about myself and my science, but before that, I'm going to talk to you about some pigs. Hi. I don't know if any of you guys have seen this in a news story. We have hands up. Who's seen this story? Yeah, I mean, it's quite a conversation starter. My friend sent me a message, because I'm a massive animal welfare advocate. She actually sent me the message, that's why, saying, oh, wasn't this awful? They just took between, these scientists in America, when it took between 100 and 200 brains of pigs, got them from the slaughterhouse, took off all the paraphernalia surrounding the brain, and perfused them with some fluids and some heaters, and God, isn't it awful? And I said, did they have the eyes attached to the brains at the time? Because the eyes are actually an outgrowth of the brain, and a lot of people forget that really they're a part of the brain. And what would be really cool is if we got some brains attached to some eyes, we put them in some scanners that can detect brain activity, and then stick a few visual stimuli with known responses in front of the eyes, and then we could see exactly what kind of results occurring in the brain of that organism without the need for a body. And she was like, don't you think the welfare of the pigs? And I was like, well, they're already dead, aren't they? I mean, but absolutely not. I'm not. But what I'm trying to say here is that you can absolutely love humanity. You can love ethics. You can be an extremely moral person, and still sometimes, as a scientist, as a person, you just leave those things at the door and you just imagine. And I think that's something that we do in science fiction a lot of the time. But they're also, it's a huge conversation starter, not just for fiction, but also for real life. And that's why I think Chimera is so powerful. Let's move back to what I actually do. So this is a picture of a chromosome. Let's all give the chromosome a wave. Hi. I love chromosomes. I think when a lot of people imagine DNA, they're imagining this long stretched out helix, and they don't actually realise how alive a molecule is. It's so alive, and it's alive in the structure that forms the chromosome, this egg-shaped structure. So it's kind of like two sausages stuck together, and in the middle there's the centromere. And the reason why it's forming this shape at the moment is because the cell's about to divide, and each of those sausages is going to be pulled apart and put into the new daughter cell. Simple. Now, some people think of this, the chromosome, when they hear of DNA, and other people think of this. A, T, G, C, T, all many thousands repeated over and over again. And you know, this isn't much of a page turner, but these are all of the books in the Wellcome collection, which I really recommend anyone to go and visit. And this is the Human Genome written out for us to have a look at. And although this is obviously hugely informative, it's kind of also not informative. Like, you're not going to get anything out of reading those books, and it's not interesting. But it is interesting if you feed it to a giant computer, and then you can start picking out exactly where the genes, the enhancers, the structural regions, and you start to see the jungle that is occurring inside the nucleus of every single cell on this planet. Next slide. Come on. Come on. There we go. So let's talk a little bit more about this jungle that I've been mentioning, because inside the nucleus, the environment isn't homogeneous. If you were to stuff your head inside the cell, stuff it right into the nucleus, you would see a bit of a hairball, but it's not just like an actual hairball. It's a very complicated, specific hairball. So some regions will have very condensed DNA that's been wound and wound and wound and really tightly packed so that it's, you know, you almost can't see it. And that's where we would tend to have the genes that are switched off. Okay, so these genes aren't being expressed. By that I mean they're not making protein that then builds the cell. And in other regions, you might have huge open loops of DNA, very easy to access for the cell. So lots of proteins can stick there, the transcriptional machinery can get there, and the genes can be expressed. Then you make proteins, and then you build the cell. So, for example, if we were to take an eye cell, in the very open regions, you would have some of the genes that were making proteins to build the eye, whereas other regions that were highly tightly bound, for example, those that make bone. I mean, no one really wants bone being made in the eye. That would be disadvantageous. So those genes are switched off, and we want that to remain the case. There we go. But like I said, DNA isn't just this long helix that's stretched out in a long line, kind of. The structure of it, the biology of it is highly specific, and I just thought I'd pick out, because there's so many different regions. We've all heard the term junk DNA. It's DNA that's non-coding, but is it still in fact a very big deal, and in fact not junk. So I thought I would just pick up on one structure, which is the very ends of the chromosomes, called the telomere. Anyone heard of the telomere? Hands up. Oh, we have some telomere fans. So the very ends of the chromosomes, and they're important because they need to be protected. We don't want them to get rusty and old. But as our cells divide, our telomeres, just the ends of our chromosomes, they get shorter and shorter and shorter. And one day they're so critically short that they signal to the whole of the cell to say it's time to stop growing, it's time to die. We have an inbuilt mechanism to tell our bodies that it's time to stop. And I think when we're thinking about immortality, it's really important to consider the telomere. It's really integral to long-lived mammals and what we can expect from our lives, how old we can expect to live. So the upshot of epigenetics, which is what I've been spending the last 5 years doing, is that every single cell is in a highly specific state, and that it's really not as simple as just dumping bits of DNA onto the cells. You can't, sometimes it is that simple actually, that's a lie. But most of the time, almost all the time, it's in such a specific state. Some of these cells, they're all doing different things. Some are dividing, some aren't dividing. And if we're going to try and mutate our genes, our DNA, we need to think about these barriers and what barriers we're going to overcome. Because at the end of the day, this is what we want to end up looking like, this lovely wee baby here. And this is not what we want to look like, which is a teratoma. And this is not a baby. It had all of the correct genetic information to be a baby, all of the genes were there, everything was ready to go, but the epigenetics was wrong in this case. So in this case, both the paternal and the maternally inherited genes had a maternal epigenetic state. If that doesn't make sense, come and talk to me afterwards. I don't have a lot of time. So we started to think about, fixing our genes over the last 30 years or so. And I just wanted to throw out one example to you about gene therapy that occurred in France about 20 years ago. So this disease called SCID, Severe Combined Immunodeficiency Disorder, just a single gene mutation, which means you basically don't have an immune system. It doesn't function. Wouldn't it be great if we could fix that one mutation in that one cell type, blood cells? And what they did is they took out the blood cells from the young kids, and they supplied them with a viral vector. And we use viruses to put bits of DNA into other cells, because that's what viruses do normally. All we do is we take out the viral DNA, throw it in the bin, and put in our piece of DNA. So the viral coat is still there, and it squirts our bit of DNA into our cells of interest that we want to transform, the cells that we want to change the DNA of. They shoved those cells back into the kids, the ones with the proper gene in there, and wow, it worked. They were making this gene. It was fantastic. They were making the protein. The immune system was recovering. But the happiness was a little bit short-lived, because within a few years, those kids had developed leukemia. And a few more years down the line, they'd all developed leukemia, and they were like, well, fuck. Wasn't ideal. And then if we start thinking about epigenetics, we start thinking about open DNA that's nice and open and free, and DNA that is very close and tight and not expressed. If we were going to put some DNA into a cell, where would we expect it to go? Even if we had the best navigation system ever, even if we had a system that could exactly pinpoint where we wanted that DNA to go, if there was a mistake, where would it go? And it would go, obviously, to the more open DNA. And in this case, the gene had inserted itself, the DNA we put in, just upstream of a gene that says grow, 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 grow. So that's what the cells did, they grew and became cancerous. So when we're thinking about putting DNA into cells, and this is something I'm always thinking about when I watch science fiction, is how we're going to get them there, how we're going to get this gene into those cells, because there's a huge number of barriers there. And in the last 5 years, CRISPR has really come a long way. CRISPR-Cas9 system, an amazing system for targeting our DNA to exactly where we want it to be. This specific targeting system, basically, we've got a guide RNA, which is key. And this, along with the Cas9 enzyme, inserts a little break in your DNA, or the DNA in your cell of interest, and the guide RNA is what takes that enzyme to exactly the right place, supposedly. After you've inserted that break, you put in your template sequence, which is then used to repair the break that you made, and hopefully to insert your gene of interest. And it's been working well. I mean, it's great and stuff. However, we've had a couple of issues, but things are still moving forward. The same thing's still basically happening. It's still going in, we're still having off-target effects, where the DNA is not going exactly where we want it to go, but it's going elsewhere. And the upshot of that is that you can't really use it for human embryos, because, I mean, even if, there's the smallest chance of going to the wrong place, it's just not an option. It's not a risk that we can take. Come on. Yes. Okay. So I've talked a little bit about getting the DNA where we want it to be if we were to make a mutation. But now I'm going to talk about what genes you might actually want to put in. Not if you wanted to fix someone, but if you perhaps wanted to augment them. So I'm talking, of course, about the Immortal Jellyfish, which plays quite a large role in the Chimera film, I'd like to think. First of all, what is a jellyfish? Is it a fish? Is it a, what is it? Is it a starfish? Sorry?
A jelly?
A jelly? Ooh, I think it probably is jelly-esque. I have poked one and it did feel like a jelly. But you never poked a jellyfish?
I'm sure.
Yeah. So basically, most of us in the room are triploblasts. I hope we all are. If we're not, we're sponges or jellyfish, actually. So triploblasts have 3 layers of tissue, embryonic tissue. Our jellyfish friends only have 2 layers, so they're diploblasts. So although they're still animals, it's not a plant, it's not fish, they're quite distantly related to us. Closer than sponges, but still quite distantly related to us. And one of the things we need to understand if we want to talk about jellyfish is their life cycle, because it's a little bit different from ours, where we know we're a small babby and then grow into a human. So that's not what jellyfish do, really. They start off life as a free-swimming, little fishy-looking thing. It looks a tiny bit like a fish, and that's quite relevant. But then it basically falls to the floor and becomes a polyp. And a polyp looks a lot more planty, still not a plant, but a bit more planty, waving itself around. And from this, the medusa bud from the polyp. And that's what you see here, is just the medusa stage of the jellyfish, the one that we all come to think about. Now, what this jellyfish can do is it can go backwards developmentally. So it can go backwards from being a medusa to being a polyp. And that means that it's switched on a developmental system and reversed it, which is quite something, a bit like a butterfly turning back into a caterpillar, but also not really much like that. So one of the things that obviously people became really interested about with the immortal jellyfish is it going to save us? Oh my God, we're all going to be immortal. But you know, this jellyfish is reversing its developmental program, and I'm not sure that's what we had in mind when we were thinking about immortality. We don't want to turn back into babies. And when you actually look at the papers that are looking at the jellyfish, they're actually studying the mitochondrial DNA, and that's quite relevant. But I won't go any further into that because it's a long, boring story. So I'm going to talk to you now about stem cells. Because as much as I love the immortal jellyfish, I'm not sure that it's going to be our saviour as a species. I think we might want to turn our eyes to the lobster or the naked mole rat, or maybe other long-lived mammals. But stem cells are really interesting, and they are, even as a scientist, you're like, ooh, yes. Because there's still something, even though it's science, there's something magical about a totipotent stem cell. We still don't really know what's going on there basically at all. So what is a stem cell? A stem cell is a cell that has the ability to turn into any cell type. That's all a stem cell is. There are different types of stem cell, and I'm talking about totipotent stem cells. And that's a stem cell that has the ability to turn into a whole organism. So some stem cells, they can turn into stuff, they can turn into lots of things, and not a whole organism. And you can take this from like the 1 or 2 cell stage of an embryo. And you can use it to do a huge amount of science with, but of course there's the ethical concerns there. You know, this could be a human. I mean, it's not, but it could be. I know we're going to throw it in the bin in 5 days, but it could be a human. So what people are doing a lot of the time now is they're making it such that that embryo, those few cells could never become a human. They're mutating them before, right at the beginning, inserting mutations so that they could never grow into a viable offspring. They could never. So they're like, well, that's fine then. I mean, it's still the same thing, but we've just, ruined it so that we can still do our experiment, but it's never going to grow into a baby. And one of the huge discoveries that Yamanaka made along with John Gurdon, which got the Nobel Prize in 2012, is how to revert some of our differentiated cells back into stem cells. And this is something else which is sort of touched on in Chimera, and that's amazing because, like I was saying earlier, your cell's in a highly specific state, and you do not want a brain cell to start thinking that it's a blood cell. But we can actually take our cells right back to being a stem cell, and that's pretty fantastic. And for me, this is incredible. This is where science is getting amazing, because it means that maybe one day we could take a cell from your own body and grow yourself a new heart with it. Because you just need to reprogram the cell, and then you need to provide a structure that you can grow those cells on, and then maybe you could regrow yourself a heart, and that would be amazing for for making chimeras without being a chimera. So you wouldn't need to borrow anything necessarily, you grow it yourself. So let's say you actually couldn't get that piece of DNA into your cells, you couldn't use those stem cells, it's not working here. What can we do? We can freeze our cells and save our cells for later. Lovely. It's tricky. At first, I have to admit, I was like, well, it can't be done, it can't be done. Of course not, it's ridiculous. And then I thought, well, we do it to eggs and sperm, and there's an Alaskan frog that does it every time it hibernates, so why can't we do it? You know, it only freezes 2/3 of its body, but it freezes itself every winter, completely frozen, and then comes back to life. The thing is with that frog, is that it has specific programs within its cells, which means that it can dump a whole load of glucose and urea into each of its cells, which changes the osmotic pressure of the cells, which means that ice crystals form in a different way, which means it doesn't die. And we can't do that, unfortunately, right now. And then it takes us right back to the issue of either needing to insert a program into each of our cells where each cell in our body knows how to produce an antifreeze, which is difficult because we have to go back to the trying to insert DNA thing, or we can just make our own antifreeze and inject it into ourselves, which is difficult because there's so many nooks and crannies in the human body, and that's basically it. You can't get that antifreeze to every single part of the body. But for me, I've been researching this for the last week, and I just don't think that this is impossible. I think that we're going to be able to do this at some stage. We will actually be able to freeze some. I'm sure everyone thinks something different based on your own research, but this doesn't seem like a huge amount of science fiction to me anymore. I think we'll be able to slow the growth of those crystals, those ice crystals somehow, so that when we bring that thing back to life, it won't be completely dead and mushy. The thing is, people are already freezing themselves. And I don't think it's a really great idea per se. This is one story about a little girl who desperately wanted to be frozen. She had terminal cancer, and she had to take her parents to court about it. The idea of consent, and particularly for me, because I'm moving into a medical career now, and I think when you think about consent, you needed to be voluntary, you needed to be informed, and you needed to be competent. So I think the idea that comes out of Chimera for me is a huge big deal, and it's something that society needs to be talking about right now. And I think it's incredibly powerful. So, I'm very happy to be here today, and I'm so excited for you guys to see this film. I think that the reason for me why this is so relevant is that we're coming through a stage now where we have technology and science beaming ahead of us, our ability to save lives beaming ahead of us, and we have our ethics and our morality kind of trailing behind, and we're kind of having to turn back and look at them retrospectively. We're at a stage where we can save a life, and we don't know if we should. And if you sit in an ethics committee and you listen to them, you're like, oh gosh, and then you've saved someone, you're like, oh gosh, was that the right thing to do? So I think it's just so powerful, and it's so important for us to be asking these questions. And also, the science is there. There is someone somewhere on this planet working on this furiously right now. And I can't stand here and say that these things aren't possible, because you always think something's impossible, and then it is so possible. So I would like to introduce Maurice Haeems' film Chimera, and I really hope you enjoy it. Thank you very much.